Okay, well, hello and welcome to today's webinar. My name's Melissa and I am the e-learning officer for the EBI training team. I also happen to be a former curator for ArrayExpress. With me today is Amy Tang, who is the curation lead for ArrayExpress. So as you can see, the topic of today's webinar is ArrayExpress, why and how to submit your data. But instead of being a step-by-step tutorial of how to submit your data, what I'm going to try and do is to get you thinking about why you need to be sharing your data, what makes a good submission, and then I'll point out a few things that can help make your submission a bit easier. So let's begin with what is ArrayExpress. So ArrayExpress is a database of functional genomics experiments. Now, functional genomics is the study of how genomics, transcriptomics, metabolomics, and proteomics come together to influence the phenotype. ArrayExpress contains data from epigenomics and genomics experiments, as well as transcriptomics experiments, which have been done either on microarrays or using RNA-seq or high-throughput sequencing technologies. So you're probably wondering, what's the point of sharing your data? Well, if your desk looks anything like mine, it's a really good way to get your records and data organized. And this will help you improve the reproducibility of your studies and make it quicker to get your data published. Of course, sharing your data is what science is all about, and it will help generate more knowledge. One thing that you might not know is that many funders and journals also require you to submit your data before you can publish it. So now we know why to submit our data. Where should we put it? Well, for functional genomics experiments, there are two databases that you can submit your experiment to. These are ArrayExpress, which is hosted by the EBI in the UK, and GEO, which is hosted by the NCBI in the US. Both of these databases take experiments from researchers, curate them, and then store them so that they're publicly accessible. ArrayExpress also imports experiments from GEO, so if you do submit your experiment to GEO, it might also end up in ArrayExpress. So what kind of stuff do we need to include when submitting our experiments? To get a better idea of this, what I want you to do is imagine you've been shown this picture and someone's asked you to reproduce this experiment. What kind of information do you need in order to do this? Well, you probably need to know a little bit about why you're doing the experiment, what's the aim, who did it originally, and what's the main experimental variable. You'll also need to know a bit about the samples, what kind of mice were used, what kind of diets, what kind of samples were taken from the mice, and exactly what kind of sequencing protocols were used. You will also need to know about the wet and dry lab protocols that were used in the experiments. So this is everything from how the mice were housed and fed the diets, right through to making the sequencing libraries and sequencing and analyzing the data. You probably also need to have a look at the original data file so that you can compare your results to the results of the original study. So all of this information is the same information that you're going to need when you're submitting your experiment to ArrayExpress. And it comes under the Miami and MinSeq guidelines, which describe the minimal information required to unambiguously describe the experiment. So now we know why we need to submit our experiments, where and what. But what about timing? When should you be submitting your experiment? So one thing that we see quite often in ArrayExpress is this scenario. Somebody will have their paper accepted and gets very excited, but then they're told that they have just 2 days to deposit it in the public database, and they panic and send us, the curators, a series of emails demanding that they have their accession number immediately. This means that the submission is being rushed, and because it's being rushed both at the curation end and by the researchers, more mistakes happen, which means we as curators need to ask you more questions, and the submission process can actually take longer before your experiment is loaded in ArrayExpress. Instead, what we suggest that you do is take a more relaxed approach and plan ahead. gather all the data and metadata, or the information about the experiment, and submit it to ArrayExpress first. Work with the curators to make sure your experiment is in the best shape possible. Then you can use the accession number that you're given in your paper, and the review and publication process will go a lot smoother. So how exactly are you going to get your data into ArrayExpress? Well, with ArrayExpress, we use a web form called Annotare, and we put a lot of effort into making this as user-friendly as possible, and we're really pleased to see that our submitters really like using Annotare. And some of the reasons they like using Annotare include that you don't need to tackle big, scary spreadsheets. There's lots of friendly curators to help you with your submission. You get your accession number quickly, and we also support double-blind peer review. So the first time you log into Annotare, you will probably come to a page that looks a little bit like this, which, if you've used it before, will have a list of the experiments that you've created. If you've never used Annotare before, then we suggest that you take a look at the Getting Started and Help pages using the buttons from the top of the screen. And these contain lots of handy information about how to fill in the form. Once you're ready to start, click on the Create button to create your experiment. But one thing to note is that if you are doing a single-cell RNA-seq experiment, some types of two-color microarrays, or your experiment has more than 1,000 samples, you should get in contact with us first, because there's a few things we need to do in Annotare to help with these types of submissions. So once we're ready to start our experiment, we click on Create, and we'll be taken to this page. And here you'll be asked, what kind of technologies did you use? Did you do a microarray or a sequencing experiment? We're also going to ask you, how many samples did you have? And it's really important to get this right at the beginning, because it's harder to go back and change it later. If you're doing a microarray experiment, we'll also ask you, which chip did you use? And to choose the chip, you just need to start writing into the text box beside this question and choose one of the options from the pop-up window. We'll also ask you, what kind of label did you use? And these are usually Sci3, Sci5, or Biotin. And you can usually find this out by reading the protocols that you use to hybridize the samples to the array. So once you've created your experiment, you'll be taken to the web form, which is divided into sections and is where you'll add all the information about your experiment. At the top of the page are the two most handy buttons, the Help button and the Contact Us button. The Help pages have lots of information about what to include in each of the sections. And if you're ever unsure about what to include, or something seems a little bit odd, just click on the Contact Us button and you'll be able to email us with any questions that you might have. So the form is separated into several sections. The first part is the Experiment Description tab, which is where you add information about the experiment and about yourself. The second tab is the Samples and Data tab, where you add information about the samples, the protocols, and upload the data files that you want to include with your experiment. The last two tabs allow you to preview all of this information as it's represented in a spreadsheet. So let's take a closer look at the Experiment Description tab. On this part of the web form, you need to give us a title for your experiment. Write a little description that includes why you're doing the experiment, an overview of the methods that you used, and any relevant background information. It's also where you choose the public release date for your experiment. But don't worry, you can change this later on if need be. We also have the Contact section and the Publication section, which is where you add information about who was involved in the experiment and how we can contact them, and any papers that are linked with the experiment. Probably the biggest part of this form that you'll need to fill in is the Samples and Data section. And the first part of this part of the form is all about the samples. It's where you describe the characteristics of the samples that you analysed. Now, the temptation with this part is to just add a big text box that contains lots of information. Although this is accurate, it's not very easy to read, both for people and for humans, and it's not possible to see at a glance what was involved in the experiment. Instead, what you need to do here is to split the characteristics of your samples into individual columns. So if we think back to our imaginary experiment, this means that we'd have an individual column for the organism, the strain of mice that we used, the organism part that we analysed, and the diets that were involved in the experiment. This is what we call sample attributes. And to add them to the form, you just need to click on the Add Sample Attributes button, choose them from the list in the pop-up menu, and then tell Annotare which of these attributes is the experimental variable. In the case of our imaginary experiment, the experimental variable is the diet. And to tell Annotare this, you just need to click the text box that is highlighted in the red square. Once you've chosen which attributes to include, you need to fill in the boxes within the form. Some of these are drop-down lists which you can choose from, and others are free text boxes. For the free text boxes, you'll need to press Enter once you've finished typing in them, otherwise your text will disappear. And this is a trap that even the curators fall into all of the time. So a couple of handy features on this page are the fill down button and the import values button. The fill down value button can be used to copy a particular value into all of the boxes below it. So for example, if you wanted to copy bulb C down for all of the other samples, you just click on bulb C, then click on the fill down values button, and it will be copied into all of the other cells. The import values button lets you copy information from Excel into the Annotare form. To use it, you need to select the cell where you want to paste it into. So for example, the diet column. Click on import values, paste the information into the pop-up window in the same order that it appears in Annotare, and then press import, and this information will be copied into the form for you. These two buttons are really handy, especially if you have a lot of samples, and it will save you a lot of time in filling in the form. So what are my hints and tips for filling in the sample annotation? Well, the first is to include as much information as possible and to state the obvious. Make sure you're including information, for example, about the strain of mouse that you used, the dose of the drug, and whether you had biological or technical replicates. You should also include information about any confounding variables, for example, the sex or gender or age of patients in clinical studies. Avoid using any context-specific acronyms because your acronyms are probably going to mean something different to my acronyms. For example, M can mean male, minutes, months, or even meters, so you can see how this could become confusing. The next section of the form is the protocols section, and as the name suggests, this is where you add information about all of the protocols you used, right from the beginning of the experiment where you're collecting samples from the mice, right through to making the libraries, sequencing them, and analysing the data. To add protocols, all you need to do is click the Add Protocols button, choose the protocols you want to include, and then fill in a detailed description of the protocols in the description boxes. These boxes look quite small, but they actually can fit in a lot of information. And the best approach with this is to write it out in Word first and then copy it into the box. And once again, don't forget to press Enter so that your text doesn't disappear. So my tips for adding protocols include as much information as possible, and know that some protocols are compulsory, for example, sample collection and nucleic acid extraction protocols. As I said, you should include as much information as possible, and if you need to, you can add attachments such as PDFs and include references in your description. For sequencing experiments, you also need to include information about who did the experiment, which sequencing facility did the sequencing, and what kind of machine did they use to do it. And last but not least, press Enter or the text will disappear. For sequencing experiments, there's an extra section on the Samples and Data tab that you need to fill in. This page is designed to give us more information about the libraries. And this is really important if you come to analysing somebody else's data, because the way the libraries that was made influences the way that you analyse the data. This information can usually be found in the protocol that you use to make the sequencing libraries. But if you're unsure of what to include, have a look at the help pages because it will explain what each of these categories means. You can always contact us as well if you're not sure of what to do. So now we come to the section where we're uploading our data. And the first thing you need to do here is check which file format you need. You can do this using the help pages or you can contact us to ask. And the file format you need will vary depending on the type of experiment you did. Wherever possible, you should also include raw data files because if somebody needs to reproduce the analysis you did, they will need to start with these files. Uploading files in Annotare is really easy, and all you need to do is drag and drop your files into this space. Alternatively, you can also use the Upload Files or FTP Upload button. Uploading files can take a little while, especially if the files are large, so you need to be patient. Once they are uploaded, you'll also need to tell Annotare which files belong to which samples. To do this, all you need to do is click on Assign Files, choose the type of data file you want to add. So this will either be a raw data file or a processed data file, and then choose the name of the file from the drop-down menu. If you have a lot of samples, you can also use the fill down and import value buttons to save you some time in the same way that we did with the sample annotation page. The final step in creating your experiment and your submission is to run a validation check. Now this check is designed to make sure that you've included all of the compulsory information. To run it, all you need to do is click on Validate, and if there's anything missing, you'll get an error message. And if you look more closely at the error messages, they will tell you what you need to include. So you need to have a read of these, go back and add any extra information, and then revalidate the experiment. If any of the error messages don't make sense, then send us an email or have a look at the help pages. Once you have no validation errors, then you are ready to submit your experiment. So now that all the validation errors have been corrected, we're ready to submit the experiment to ArrayExpress. So to do this, it's simply just pressing the big green button at the top of the page. You can also download your submission as a spreadsheet at this point. Once you've done this, you'll soon receive an accession number, but this doesn't mean that the submission is complete. The next step is for a curator to check over your submission and make sure that it's in the best shape possible. And if they have any questions, they'll email you. Once they're satisfied, they'll upload your experiment into ArrayExpress, where it will be kept private until it is published or you tell us to release it. Once it's in ArrayExpress, it's given a page that looks a little bit like this, and which contains links through to all of the information that you've just added in your Annotare form. At this point, you're no longer able to make changes using Annotare. So if you do want to add details about your paper, change the release date, you've spotted a typo, or you want to add more samples, you will need to contact us in order to do this. But wherever possible, we encourage you to make sure that your submission is complete as possible before you submit it, because making changes is more difficult once it's loaded into ArrayExpress. So we've finally made it through the submission process and our experiment is in ArrayExpress. So just to summarize, what are my top tips for submitting your data to ArrayExpress? So know that submitting your data generates knowledge and it's often required by funders and journals in order for you to publish it. You should always start the process early. It takes time, and the more effort you put into it, the better quality your experiment will be. Include as much information as possible, even if you think it is obvious. Try and think about what you would need in order to reproduce the experiment. Avoid using acronyms or shorthand and write things out in full. Acronyms that I use probably mean something different when you use them. And last but not least, ask for help. There are always curators on the other end of the line, and we're more than happy to help you with your submission. That is, after all, what we're there for. So just in case you missed it, if you ever need help with submitting your experiment to ArrayExpress using Annotare, you could use the Help button at the top of the page or contact us using the Contact Us button. Alternatively, you can email arrayexpress@ebi.ac.uk. And if you want to know more about ArrayExpress, you can try some of our courses online in Train Online using the link below. So finally, I would just like to acknowledge and thank all of the people behind ArrayExpress, including the curators and bioinformaticians, the data management team, and the web development team. So that's all we have time for today. Our next webinar is coming up on the 5th of October and will be about training opportunities at the EMBL-EBI. You can see the full list of upcoming webinars at the URL shown below. Thanks again for joining us, and we hope to see you again soon.